Your brain is extremely greedy. Okay, hold on. Before you switch to a less offensive video, let me explain. Your brain is only about 1 to 3% of your body weight, depending on how heavy you are. Yet it uses about 20% of the energy that you take in from food every day. Now, 50% of that energy, so 10% of the total energy you take in, goes exclusively to one single type of molecular machine that is used in neurons, and we'll talk about that later in this video. But the question is, why does your brain use so much energy, and why does that little machine use so much of the total energy that your brain uses? The answer is that it comes down to the fundamental mechanism by which your brain processes information, and that is called the action potential. That's going to be the subject of today's video, and by the end, you will be able to understand how scientists are able to monitor the activity of small populations of neurons, as well as how Elon Musk's new revolutionary technology, Neuralink, works, as well as a scary sounding but actually very effective treatment for major depression called electroconvulsive therapy. So let's get into this. Okay, so before we get into the nitty-gritty of the science, you might be wondering how learning about the brain on this fine-grained level of detail can tell you anything about the mind. But interestingly, there has been a rich history of neuroscience research where scientists are basically recording the activity of very small populations of neurons, and they're able to correlate that to certain behaviors. So for example, a 2019 study published in Nature found that a collection of just 31 neurons in an area at the front of the brain called the dorsolateral prefrontal cortex were able to predict how a subject would make a decision. So more specifically, these subjects were told to look at a series of images and decide whether they looked like they were safe or not. So for example, it might be somebody riding on a bicycle in the street with or without a helmet, and you as the subject would have to decide whether you think that is safe or not. So by recording from just these 31 neurons in the front of the brain, the scientists were able to predict whether a subject would decide that that scene was safe or not. And basically, they could see that the activity of those neurons, about half of them would turn up when the subject decided that it was a safe looking scene, and the other half would turn down, and the opposite was true for the unsafe scene. So the reality is that there's many more than just these 31 or so neurons involved in the decision-making process. But the point is that by just pinpointing this little part of the network that's involved in that decision-making process and monitoring the activity of this small population of neurons, the scientists were able to understand at least something of what was going on in the brain during this decision-making process. So, you might be listening to all these terms like activity and neurons firing and that sort of thing and wondering what that is, and that is the subject of today's video. So before we dive into action potentials and how neurons communicate, I want to do some introductions. I'm Andrew Cooper Sansone, and this is Sense of Mind. This channel is all about giving you the tools so that you can understand your brain, upgrade your mind, and improve your life. The idea is to give you fundamental knowledge about neuroscience and psychology, as well as insights from elsewhere in the world of science and understanding, and to allow you to apply those to your life so that you can make decisions that let you live a better life. The video you're watching right now is part of an ongoing series that is basically focused on giving an introduction to neuroscience to anybody who's interested in how their brain and mind work. We will be going into some biology as well as psychology. We'll be kind of spanning the whole spectrum, but we're starting with the small, starting with neurons and building outward. So today's episode is about action potentials. Now, you may have heard this term before when someone refers to a neuron firing or spiking or even just turning on or activating. These are all terms for the same thing. An action potential is the process that a neuron undergoes in order to communicate with other neurons. Now, an action potential is actually an electrical event. We'll talk about how that works. But the actual communication between neurons is chemical. They're basically transferring chemicals between each other. So the action potential is actually what allows for those chemicals to be released. And the release of those chemicals and the binding to the next neuron is what we'll talk about in the next set of videos. This video is specifically focused on how an action potential occurs. By the way, if you don't know what neurons are or you're not familiar with their basic anatomy or what they do, check out the earlier videos, the first set of videos in this series. We have 3 different levels of difficulty. And that will also be true for this video. So for every video in our Introduction to Neuroscience series, there will be a 30-second summary of the most important point about the topic, as well as a 5 to 10-minute video that will basically explain it in a little bit more detail, but not really get into the technicals and nitty-gritty. And then we'll have a longer video like this one that goes fully into depth and explains in full detail. So at whatever level you are comfortable with or that you're interested in learning about, we will have a video for you. Okay, so this is your brain. Now, if we were to zoom in on just a tiny, tiny little patch, in fact, it's much tinier than that square would make it look, but let's just imagine zooming as far into the brain as we possibly can and finding a single neuron. Okay, so this is a neuron, and just as a quick review, I'm going to go over the big different anatomical parts of a neuron. So first we have this sort of central area called the cell body, and I have receiver written there because it can receive neurotransmitters. We'll talk about that in a second, but anyway, the cell body, this is where the DNA is housed. This is where a lot of important processing of information is going on. It is kind of the heart of the cell. Then we have the axon, which we can consider a sender. This is the cable, kind of the arm, the mouth of the neuron. It allows the neuron to communicate with other neurons. And we finally have the dendrites. These are the receivers. These basically communicate, they synapse, they connect with other neurons, and they listen to what those neurons have to say. So that is the basic morphology of a neuron. Now keep in mind there are many different types, and so some will have fewer dendrites or more. Some will actually have more than one axon, but this gives you an idea of what a neuron is. And to be totally clear, this totally underestimates how many connections a neuron has to others, because it just looks like, you could count them. There's about maybe a dozen dendrites there, but the reality is that you zoom in and dendrites have what are called dendritic spines, and they connect. So a neuron can connect with thousands of other neurons, which is pretty amazing. Okay, so the very first thing that you have to understand is that a neuron is negative. By that I mean it is negatively charged relative to the outside environment. So as you can see in this graph, that corresponds to this initial flat line near the left side of the x-axis. This is called the resting potential because it's the voltage at which a neuron is usually when it's just resting and not doing anything, and it's typically around negative 70 millivolts, although that can vary depending on the type of neuron that we're talking about. So you might wonder, why would the resting potential of a neuron be negative? What causes that to occur? So one big reason is that the concentration of positively charged ions inside and outside of the cell is different. So ions are basically just charged atoms or molecules. And so, you can imagine if you have this neuron and it's filled up with positively charged ions, it would have a positive resting potential. It would be positively electrically charged relative to the outside environment. But instead, what usually happens is that neurons have a very low concentration of positively charged ions, specifically sodium ions. We'll talk about the different ions that are involved. Okay, so now we're still looking at this neuron, and I want to draw your attention to just one of these dendritic tips. So let's go into the dendrite and see what's going on. Okay, so this, I put dendrite or cell body because, as I mentioned, they can both receive neurotransmitters, and it doesn't really matter for our purposes which one is which. This big green box is the dendrite or the cell body, and the black line, the border surrounding it, is the cell membrane, so that's what separates the cell from the outside world. And then this orange thing we have here is a neurotransmitter receptor. Now at the bottom right of the cell, you see that there's a voltage, and we're going to be tracking the neuron's voltage throughout this little slideshow, and that will allow you to see how it changes as time goes on. Okay, so right now we're at the resting potential, right? -70 millivolts, everything's cool, nothing too crazy going on. All right, so now what we see is a neurotransmitter coming into the picture, right? And this is just a molecule. This could be glutamate, GABA, dopamine. It doesn't really matter here for our purposes, but it is a molecule that binds to a receptor. And okay, so that neurotransmitter binds to the receptor, and then what happens is these red circles and the arrow represent a chemical reaction, basically a chain of chemical reactions that occurs after that neurotransmitter binds to the receptor. It's a kind of cascade of molecular events that occurs, and what you're seeing is that those little circles are representing little chemical reactions going on, and what happens is they interact with something called a sodium ion channel. And so the sodium ion channel is that black tube right there, and connected to it is just that little red Pac-Man guy. That's just showing that there's a chemical reaction happening that basically causes that sodium ion channel to be able to allow sodium through. So it's called a sodium ion channel. I mentioned this earlier because it only allows sodium through. It's selective, right? And it only will allow it through once that chemical reaction has occurred. So the receptor binds the neurotransmitter, which causes a chemical reaction inside the cell, which then kind of goes through a chain of events that ends with another chemical reaction happening on the sodium ion channel, which allows it to open up and sodium to flow through. So sodium flows through, right? And what happens there is it changes the voltage, the potential of the cell from -70 to -55 millivolts. So it goes up. That's a rise in voltage, becoming less negative. All right, so that is what happens at the dendrite or at the cell body. Now, the next thing to happen, the threshold value, that -55 millivolts, has to be reached at the axon hillock. So let's just look again at our neuron to get a sense of what we're talking about. So that arrow, that big black arrow, is pointing to an area where the axon hillock would be on this neuron. So it's right at the base of the axon. And you can kind of imagine why the action potential has to be initiated there, because after all, you do send neurotransmitters with the axon. So at some point, it's got the action potential, the change in voltage has to reach the axon, and it makes sense that it would occur right at the base. So basically what's going on is this neuron is receiving input. It's binding neurotransmitters like all over the place on its cell body, on these various dendrites, and all of these are causing these tiny changes in voltage, and those are kind of all adding up, and eventually that region of the axon hillock reaches the threshold value of about negative 55 millivolts. So that is when the action potential really gets underway. So what we're seeing in this image is a depiction of the axon hillock on the left and then the rest of the axon on the right. So now we're looking at these voltage-gated sodium ion channels, and I mentioned those in the description earlier. But basically what these are the same sort of tunnels that only allow sodium into the cell, and they are only opened, instead of being opened by a chemical reaction, they're only opened when the voltage reaches that threshold value. So the axon hillock reaches its negative 55 millivolt threshold value thanks to all those voltage changes happening elsewhere in the cell and then adding up at the axon hillock. So what happens here is sodium ion channels are triggered to open because of that change in voltage, and this allows sodium to flow into the cell, basically. So all the sodium flows in, and then what happens is that kind of causes the membrane potential, the voltage of the cell, to go way up, way higher than the negative 55 millivolts. It in fact goes to the positive range into plus 40 millivolts. So that actually causes those sodium ion channels to close because they're voltage sensitive, so they're only open within a certain range. And now technically it's a little bit more complicated than just closing. There are different stages of deactivation and closing of the sodium ion channels. We're not going to go into that because basically what they do is stop allowing sodium through. And what happens is all these little sodium ions, these Na plus ions, diffuse into the rest of the axon, right? Because the rest of the axon is still at that negative 70 millivolt resting potential. It hasn't had any sodium enter into it. So now the sodium begins to diffuse across into the axon. And what happens there? Well, it's just the same thing, right? The voltage goes up, and that triggers those sodium ion channels in that slice of the axon to open up and allow more sodium through, right? And then again, it causes those to close because they get to a high enough voltage that they close, but those sodium ions are still flowing down the axon, still diffusing down their concentration gradient. And again, this goes on and on down the axon at 340 miles per hour. This is traveling really fast down this really small distance. And it's going down that axon, and eventually you get to the tip of the axon. And like I mentioned, this is where the release of neurotransmitters is triggered. So you have this wave of basically of changing electrical charge of the neuron becoming positive, or the axon becoming positive in sort of sequential adjacent slices. As it's moving down the axon, is becoming more positive. And eventually it causes this neurotransmitter package to be released. Now we will talk more about exactly how that works in the next few videos, and how drugs affect that process, how they can mimic neurotransmitters. We will get into all of that. So that neurotransmitter leaves and it goes to bind onto the next cell. Now, we are left with a very weird situation where the voltage is super high and there is a lot of sodium in the cell. This is basically the opposite of the resting potential situation for a neuron. Now, something I didn't mention is that inside the neuron, there's usually a very high concentration of potassium ions. So potassium I have here as K+, that's its symbol on the periodic table, and Na+ is sodium symbol on the periodic table. Regardless, so potassium is very high inside the cell. So now we have this situation where there's tons of potassium, tons of sodium, and it is a positively charged voltage inside the cell. What happens here is that when we reach that super high voltage, not only does it cause those sodium channels to stop working, to close, it causes these potassium ion channels to open. So that allows all this potassium to flow out of the cell, right? And the result of that, of course, is to lower the voltage because we're releasing positive charge outside of the cell. So now the voltage actually goes way down into the refractory period where it's below the resting potential. It's at about negative 75 millivolts instead of negative 70. And that situation sort of resolves itself. It gets back to negative 70 millivolts. And now we're left with that problem where we have tons of sodium inside the cell and tons of potassium outside of the cell. We're at the right voltage, but we need to reestablish that concentration differential. So how do we do that? I mentioned the sodium-potassium pumps. And this is a depiction of one. It's obviously nothing like what it looks like in real life, but it shows the important workings of this machine. And in fact, it's really not all that different how it actually works. But what you can see here is there's kind of two arms to this machine inside that blue area. And one arm has two slots on it, and the other has three. And now the three-slotted side is attracted to sodium. So sodium will naturally flow into this pump, and 3 sodium ions will bind into the 3 slots on the pump, on the 3-arm side. So then what happens is this molecule, this sodium-potassium pump binds a molecule of ATP. And as we mentioned, ATP is the molecular battery pack. It is the energy currency of the cell. And that causes a chemical reaction that releases energy. And what that does is allow the sodium-potassium pump to change its conformation, change how it's arranged, change its shape. So it opens up to the other direction. And that causes the sodium to be released and to flow outside of the cell. Then what happens is that potassium ions are attracted to the 2-slot arm, the other arm of the sodium-potassium pump, and they will flow in, bind to that side of the sodium-potassium pump, and it will just naturally reset itself. And that will cause the sodium-potassium pump to open up to the other direction, so toward the inside of the cell. And those potassium ions will flow out into the cell. And this process just repeats itself over and over again until what you've got is lots of potassium inside the cell, a little bit of sodium inside the cell, lots of sodium outside the cell, a little bit of potassium outside the cell. So now this is why your brain uses so much energy. This takes a lot of energy to do this, to reestablish that ion concentration gradient. And so like just to give you a sense of this, if you ate a banana, say you just ate a banana, and you imagine that banana cut into 10 equal pieces, right? You take 2 of those pieces, and those are just for your brain. The rest is for your body. But one of those pieces is just for these sodium-potassium pumps. And so that's that's pretty amazing. There's a lot of other processes going on inside neurons, inside other cells of the brain, and yet fully half the energy that the brain uses are just for this one process. So we're back at our original situation. We have fully reset both the voltage, the membrane potential, and the concentration gradient of the ions. So that is the detailed explanation of how an action potential works. Now you are in a position to understand all those things that I mentioned at the beginning. That study that looked at just those 31 neurons, as well as Elon Musk's Neuralink and electroconvulsive therapy. So you won't fully understand every little bit of them, but now that you understand the electrical basis of this mechanism, this fundamental mechanism in the brain, you'll be able to understand the sort of the fundamentals of how those experiments and technologies work. So to understand that study I mentioned and the Neuralink technology, really what you have to understand is something called single unit recording. So this is basically where electrodes, microelectrodes, really thin wires basically, are inserted into the brain, and they can be used to record the changes in voltage, so basically the activity of small populations of neurons. And that's really all they're doing is measuring the changes in voltage, and so electric charge basically. And with Neuralink, what is going on is they're inserting not just one microelectrode, but thousands. And these are then used to basically record the activity of neurons that are involved in many different processes in the brain, and then they can decode that activity. So, for example, one of the future applications, kind of a revolutionary application of this kind of brain-machine interface, will basically be to allow patients who have suffered a spinal cord injury and can no longer walk, or are quadriplegic from that same sort of injury, to walk again. Because the thing about paralysis due to spinal cord injury is that the muscles and the nerves throughout the body still work, but it's just that the spinal cord, which is basically the information superhighway from the brain to the body, is broken. So it can't transmit that information. So what Neuralink and other similar technologies do is they would be able to essentially record the patterns of activity from areas of the brain involved in moving the body, moving the legs and arms, and then decode that activity, and then send that to another device that would communicate with the nerves and muscles and allow that person to move their body. So another slightly less revolutionary application of the Neuralink technology would be the Neuralink app, which would basically allow you to control your phone with your thoughts. Okay, so before we go, I want to give you some practical advice that you can walk away with. This may only apply to some people, but I think it's important for all of us to know about. Okay, so if you or anyone you know is struggling with major depression, major depressive disorder, and you or they haven't responded to antidepressants and psychotherapy, which are kind of the first line treatments for that disorder, and if nothing else seems to be working, your doctor might recommend, or you might want to ask your doctor about electroconvulsive therapy. So it sounds pretty scary, and the goal of it sounds even scarier, which is basically to induce a seizure in your brain. So that probably sounds pretty weird, and you might not know what a seizure is, but now that you understand an action potential, all a seizure is basically a synchronized wave of action potentials moving across the brain. So that seems like it might not do anything for depression. It would just give you a seizure and then return you to your normal state, right? But what scientists found is that more often than not, people respond very well, although the relief that they experience is often short-lived. So scientists really aren't certain about the mechanism that is allowing this to work and why some people's brains respond to it and why others don't. But, you can kind of think of it like it's sort of like a reset of your brain in some ways. It's just this wave of activity that just flashes through the brain, and perhaps there's some circuits that needed to be activated to allow a depressed person to feel better. And so maybe for some people that's just what they need, and maybe it doesn't last forever because those circuits go back into a more quiescent state. This is all speculation, but the fact is that electroconvulsive therapy does work for very severe cases of depression. And of course, never try shocking your brain on your own, no matter how depressed you are. Talk to your doctor about this kind of thing. It is really only suitable for a small number of people. So just be aware of that. But it is kind of a last-ditch effort that can help some people. There are other forms of electroconvulsive therapy that don't involve just placing electrodes on the scalp and pulsing electricity through the brain. There are others that use transcranial magnetic stimulation, which is basically using oscillating magnetic fields directed at specific areas of the brain instead of an electrical current, but the idea is the same, to induce action potentials or to suppress them in certain areas of the brain. So that is kind of the basics of how electroconvulsive therapy works. All right, so now you understand the fundamental mechanism behind information processing in the brain. You know why your brain uses so much energy, what the sodium-potassium pump is, and why it uses half of the energy that your brain uses. You understand how an action potential works, that it's basically a rise in voltage that causes a release of neurotransmitters, but you also understand the specific, intricate mechanism by which that occurs. So in the next video, we're going to be moving along a little bit more and talking about how the process of releasing that neurotransmitter occurs and how it binds to the next neuron. And that will get us into a conversation about how drugs affect neurotransmitter systems and, a lot of different hot button things. We'll talk about these neurotransmitters like dopamine, glutamate, serotonin, words you've probably heard before. And yeah, we'll get into drugs like alcohol, marijuana, cocaine. We'll talk about how they work in the brain and why, you might or might not want to avoid doing them. Okay. Also, be on the lookout. We are having videos that aren't a part of this series come out as well. And those are much less technical, much more focused on just purely kind of giving you the tactics that will help you live a better life, that are based on both science and my personal experience. So those will be coming out kind of interspersed between these episodes of the Introduction to Neuroscience series. So if you don't want to miss any of those videos, be sure to click the red subscribe button and also like this video, and also hit that bell icon next to the subscribe button so you never miss a new video from Sense of Mind. Be sure to follow us on Instagram and Facebook and sign up for the newsletter. The links for all of those are found in the description below this video. Thank you so much for watching. This channel was brought to you by the Diamond Mind Foundation. It was written and produced by me, Andrew Cooper Sansone. Thank you so much for watching. I'll catch you next time.